Today we're going to talk about the eye. We're going to talk about structures through which light passes, which is the cornea, the lens, and the transparent portion of the retina. We will also talk about structures that modify the image, and that would be the iris, which regulates the light going into the eye, as well as the ciliary processes and ciliary body and ciliary muscle that contracts to regulate focus of the lens. So if you find this information useful, please consider subscribing to YouTube and sharing it with your friends. Thank you. Medical School Histology Basics, the eye. Hi, I am Larry Johnson. I'm a professor at Texas A&M University. Today we're going to talk about the eye. We want to cover the things through which light passes, which is the cornea, the lens, and the transparent portion of the retina. And we're going to talk about things that influence the image, which is the iris, regulating the amount of light coming through, the ciliary bodies, and the trabecular meshwork. And here we can see the eye. We see the major globe of the eye, the vitreous body inside there, and we can see a couple of three layers that we have there, composed of different layers. Cornea, lens. Between the eye and iris is the posterior chamber, anterior chambers is at the surface up through there. And it's aqueous humor that's in the anterior and posterior chamber, vitreous humor in there. Now, if you know the workings of a camera, then you already know the workings of the eye. The shutter would be the eyelid. The diaphragm would be the iris. The lens is the lens. The sclera is the white of the eye, or the structural part of the camera. The choroid is the blackened box on the inside of the camera. You've got black to prevent backscatter of light. And then the photosensitive film would be the retina. And in fact, there's three layers of the eye. There's a supporting layer, which is the sclera, the white of the eye, and the cornea, and that makes a complete globe. That then pressure is exerted to the eye to keep the globe open, kind of like a basketball. It's pumped up with pressure. But the secretion of the eye is about twice that of the eye for a day, for the total volume of the eye for a day, and so therefore you have to regulate the outflow, and we'll talk about how that occurs. The next layer is the vascular layer, and that is your choroid, as well as the ciliary processes and the iris itself. All of those are vascular layer. We call that a uvula. And then there is a retinal layer. It comes up from the optic nerve, and it comes around here all the way up to here, where it no longer is photosensitive, but it continues all through here, and is actually part of the iris itself. So the pigment and the dilator muscle are all part of the retina themselves. So we have the cornea, the iris, ciliary bodies, lens, vitreous body, as we saw a while ago. Now the main part of the eye, of course, is the retina for photoreception. And here we see the rods and cones, pigmented epithelium, bipolar cells, and the ganglion cells, the different layers that make it. And so the light comes in through here and projects up through there, and finally it hits the photosensitive portion of photoreceptor cells, which are here. Here's our nuclei, here's some of their cytoplasm, but the photoreceptor cells portion of them is right down in there. And then you have the pigmented epithelium. Below that you have the choroid, the rich vascular supply. So we'll learn all those words. So the conjunctiva is the stratified squamous epithelium that's on the inside of the eye, and also it covers the white part of the eye itself. Stratified squamous epithelium, non-keratinized because it's basically in a liquid state. And then the sclera is the white of the eye that we see here. So the conjunctiva is right over through there, and that's what we see in through there. So whenever the cornea was developing, here's the cornea here, the epithelium from the skin, the stratified squamous epithelium from the skin, came in through there and made the conjunctiva surrounding the inside of the eyelid, as well as covering the white of the eye as well. And so then we still have stratified squamous epithelium at the top of the cornea, as we'll see in a minute. So in terms of looking at a real eye, we have the sclera here, this heavy band of connective tissue, the retina in through here. Right between the retina and the sclera, you have the choroid layer, and it's right in through there, right above the sclera, all in through there, and that's a rich vascular supply that provides nutrients to the retina. Then we have the lens, and this is where the pupil would be, the opening there between the iris. Actually, iris goes all the way around and makes a circle. But we just see two projections of the iris, the posterior chamber, anterior chamber, lens, iris, ciliary bodies, sclera, as you can see, retina, the different portions. If you look at the back of a monkey's eye, we see the optic nerve coming in through there, bringing blood vessels in through a nourishment of the retina itself, and then the different layers of the retina, including the choroid. The dark part here is the choroid. Remember the blackened box, and also it has a rich vascular supply, some nerves out through there, but that's basically the structure that we will see. If we look at the cornea, which is stratified squamous epithelium at the surface, Bowman's membrane, the little membrane through there, and then you have the stroma cells, the keratocytes, or the fibroblastic-like cells that you see there, and they're sparse. The nuclei are far apart because there's a lot of fibers between them. And below that we have a descemetous membrane, which is the basement membrane of these transport endothelial cells. Usually they're cuboidal or somewhat squamous, mostly cuboidal, low cuboidal epithelial cells. So we have 5 basic layers from the inside, from the outside to the inside of the cornea. And we can see those here. If you see the stratified squamous epithelium on the surface, Bowman's membrane here, and then you see the stroma, and the stroma has corneal fibroblasts. You can see the nuclei of these cells, you can see the cytoplasm of this cell, and there's a lot of fibers in between, and those fibers are arranged perpendicular to one another so that it selects out desirable light rays. So you have stroma from there to Bowman's descemetous membrane, which is a basement membrane of these transport endothelial cells that are on the surface, the anterior surface. The anterior chamber surface is really the posterior portion of the cornea that you can see there. Stratified squamous epithelium is what we see there. Stratified squamous epithelium continues with the conjunctiva and through the Bowman membrane, stroma, descemetous membrane, cornea, endothelium. This is endothelium, not epithelium, endothelium. As we can see there, here's the iris, anterior chamber, posterior chamber, and the aqueous humor is the liquid that's in both the posterior and anterior chamber. It's actually produced by these ciliary processes. The secretory portion goes around the iris and then is removed up in this region, as we'll see. Here's a ciliary muscle in through there, which is located located all the way around, of course, but we just see it on either side, right where the iris comes into play, is attached. In the ciliary muscle, we can see it's a smooth muscle. We can see nerves. We can see blood vessels of the ciliary muscle, and the ciliary muscle is what contracts to change the shape of the lens. And also here we see the ciliary processes. These processes, you have secretory epithelium on the outside and pigment epithelium on the inside of both these images, and then there's the lens. Iris pupa is an opening in through there. This space here is artifact. This shouldn't be. You should have no space here. Ciliary muscle, and this is where the sclera comes in contact with the cornea. Cornea has no blood vessels in it because that would interfere with light coming through. And right where you start picking up these blood vessels here is the sclera cornea junction, and that's known as a limbus. important region in through there. And right below there is trabecular meshwork and the canal of Schlemm. And so here we can see the ciliary processes that are in the anterior and the posterior chamber, and they have secretory epithelium on the outside and pigmented below there, as you can see, and we'll understand that here a little bit later. But the pigmented epithelial cells become a dilated muscle under the iris, and the secretory cells become pigmented under the iris. So you have a double layer that comes in from the retina, and they continue to be double layers, but they have different roles when they hit the iris. Now, we mentioned that endothelial cells here as opposed to epithelial cells out on the outside. So these endothelial cells, which are really endothelial cells, and they're continuous with the endothelium of the trabecular meshwork. And trabecular meshwork actually comes all the way in through there. And then, so fluid comes through there, goes through the trabecular meshwork, goes through the connective tissue, and goes through the endothelial cells with canal Schlemm to get out. And that is the outflow of the fluid. They're produced over ciliary processes. The secretory epithelium secretes it. Fluid migrates over through the pupil, and then ultimately is outflow. But the trabecular meshwork is really resistant to the outflow. If there was a direct connection with the canal Schlemm between the anterior chamber and canal Schlemm, there'd be no pressure. And so it maintains a little bit of pressure by uniform pressure by regulating the outflow, and it resists the outflow. So there will be some pressure built in the eye, but it doesn't prevent it. It just resists as a main function. But endothelial cells here are continuous with endothelial cells of the trabecular meshwork. Here again we see the trabecular meshwork in through here, little openings of these endothelial cell lined spaces, and these spaces here are continuous. The lumen is continuous with the lumen of the anterior chambers, where you have the aqueous humor being absorbed in through here and ultimately going back to the bloodstream via the canal of Schlemm with no lymphatic in the way. Here we see the ciliary muscle, the limbus region. We're right in this region, right in here. There's a lens, a cornea, as we can see. Here's a canal of Schlemm, trabecular meshwork on another view. This is the iris, and the iris here we can see it has dilated muscle in through there, and it has pigmented cells. Blood vessels, remember the iris was part of the vascular area layer of the eye, and it has blood vessels inside there. Have to have continuous capillaries to prevent antigens from going in through there, because the aqueous, there's a blood aqueous barrier. So blood-borne components have a hard time getting through. We don't want them actually into the liquid of the eye. Ciliary processes, as I mentioned, you have secretory cells on the outside, and then they become pigmented on the iris. The pigmented layer of the ciliary processes become the dilated muscle, and so we have dilated muscle. Dilated muscle is myoepithelium. It's not smooth muscle, it's myoepithelium. And here we can see the dilated muscle coming down through here, pigmented epithelium coming in through here, and then here we can see the constrictor muscle. So this is a constrictor muscle. This is the posterior chamber and the anterior chamber. Oddly enough, there is no epithelial lining of the anterior portion of the iris. So we have the pupil, the opening in through the iris in through here is what we're seeing in through here. Lens capsule, and on the anterior portion of the lens you have these little cuboidal cells, which ultimately in the bow region give rise to the lens fibers. And so here we can see, as I mentioned to you before, the cornea developing, and then also part of the retina coming up through there, and it's actually part of the iris itself. Here we can see it in the drawing where the retina comes in. There is your optic nerve coming in there, your retina. So optic nerve, retina, and then those two layers continue on underneath the iris, and are the dilated muscle, and the dilated muscle and the pigmented layer that prevents the backscatter of light. So two main functions of this regulating light is actually coming from the optic nerve. Lens, as we can see. Here we can see the constrictor is actually smooth muscle cells as opposed to the dilated muscle, which is myoepithelium. On the lens, we can see a nice cube cells here through here, and the lens capsule. You can see the capsule is quite big on the anterior surface. So if we look at the lens, here we see the lens coming in through here, actually developed before the cornea did because it got to gain access to the outside, so it's from the outside. And see how it folds back on itself. When it folds back on itself, then these little cube cells of the lens are in the anterior surface, not in the posterior surface. And that's why you have these on the anterior surface in the adult eye. As you see, you do not have them on the posterior surface because it's right in the bow region that these cube cells give rise to the fibers which run along the path of the light. So as you have the infolding in through there and back, really the base is on the outside, and such that the capsule itself is really the basement membrane. It's a shared basement membrane of these cells. It's thicker on the anterior surface than the posterior surface, as you will see. So if we look at the cube cells, there are these cells here, what got folded back on itself as through there, and the capsule is the base of those. So this is the apex, that's the base of these this particular cell. So this is the anterior portion of the lens, and the posterior portion of the lens has a fairly thin capsule, not thick like we see there. And so here we're seeing this is the capsule of the base of the back, as we can see, and the capsule is shared a basement membrane of the lens fibers. And so lens fibers, which is what we see here, in this case, is the basement membrane because the things are flipped, as I mentioned to you. So here we see the iris again, the pigmented layer, the constrictor muscle, dilator muscle, the ciliary processes, and these are ropes or ligaments or zonules. And if we look at the lens again, we can see the anterior surface. You have the pigmented, the cuboidal epithelial cells, and a thick capsule, as we see here and here. So this is the lens cuboidal epithelium that is on the surface of them. Right in the bow region, right where it turns here like a bow and arrow, the bow region, these little cube cells, which you see here and here, give rise to the lens fibers. You can see where the cells are elongated, and they become the lens fibers themselves. And so the lens fibers run like this, which is down to which the light goes straight down to fibers. Limb cuboidal cells give rise to the, and here we can see the capsule. The capsule, you can see just a little bit of it, very thick in through there. And so here we can see the lens. There's the anterior surface, which has the cuboidal cells. The posterior surface do not. Both of them have capsules, and also both of them attach to the zonules. These are ligaments or zonules, which upon contraction of the ciliary muscle, they pull or loosen pressure on the lens itself. And here you can see where one of those zonules is attaching to the surface of the lens. There's a hieroglyph over here, a zonule attaching to the capsule on this anterior surface of the lens. You can see it over here too. Another one over there, you can see where the zonule is actually attaching to the capsule. It pulls on the capsule, and it changes the shape of the lens. When the ciliary muscle contracts, it loosens the pressures on the zonules, and so when it does that, the lens goes to a more spherical shape, and that's what it wants for close vision. If you're hiking, looking at a distance, then your ciliary muscle is not contracted, and that pulls more on the zonules, which makes the lens more flat. So it's actually changes the shape of the lens from what it wants to be. It wants to be more round. If you look at the posterior surface of a lens, you can see that you don't have those cuboidal epithelial cells, and also the shared capsule or basement membrane of these fibers is just not as thick. And we can see that again here. This is the capsule of the anterior surface and the capsule of the posterior surface that you can see there. So here's the zonules, and note that the zonules come in through all the way down here to the ora serrata, which is a region that no longer is photosensitive. It doesn't have photoreceptor cells. So this is continuous with the retina. The pigmented layer here is continuous with the pigmented layer of the retina that we will learn about. So here's the sclera, choroid, ciliary bodies, ciliary processes, ciliary muscle, as we can see, and this collectively is a ciliary body. So here we can see at the other end is the optic nerve coming in, and giving rise to the retina on either side. And so we have the retina here. There's no photoreceptor cells here. That's why you have a blind spot. Pigmented epithelial cells, you can see them right in through there, right in through there. More hieromag, this is one cell, there's a nucleus, there's another. You can see the pigmented layers that you see there, the combs and the rods, and the nuclei of the photoreceptor cells and the pigmented cells, and this is a choroid. The choroid is right in through there, elaborate blood vessels of the choroid plexus that is in there, as we can see. And so basically there's several layers of the retina, but we'll just mention a few. We'll mention the retina is composed, as far as our purposes, the ganglion cells, the bipolar cells, the photoreceptor cells, and the pigmented epithelium, and the choroid below. So we can see those. There's a vitreous body up through here. The light would be coming down in through here. This is the ganglion cells. That's the one that talks to the brain. Bipolar cells. This space here is artifact. Shouldn't be there. These are the cell bodies of the photoreceptor cells, the rods and combs, as you can see. And this is a pigmented layer. The pigmented layer here is the same as the pigmented layer we see there. But that's the photosensitive portion of the rods and combs is right in through there. See the rich vascular supply of the choroid and the sclera. So we can see the ones that are rods. These are the nuclei of these cells, photoreceptor portion of them, pigmented epithelium there, combs look like ice cream cone, and the rods is what we see. So the optic nerve is coming in through here, as you see, and then here's the retina. The retina over here, pigmented epithelium. You should not have the space in through there. These guys actually touch in through there. But the pigmented epithelium, you can see it like this is one cell. There's another cell. Here's its nucleus, and it has melanin granules. The melanin granules prevent the backscatter of light. So any light that goes through there is absorbed and doesn't defocus the image or disturb the image because because it is absorbed. Now, if this was a nocturnal animal, it would not reflect light. It would allow reflect light because it would not be pigmented in certain areas. That's what's reflecting back from the eye, is that you're seeing through there. And there's actually another connective tissue, tapetum lucidum, right in through there. We don't see it in humans, but it'd be another little layer in through there that reflects back to let a lion be able to see at night. Not well, but better than not seeing at all. Choroid plexus, sclera. You can see blood vessels in the retina. Rods and combs, we can see the optic nerve coming in through there. These stretches here really artifactual space that shouldn't be there. You can also see capillaries in through there too. Photoreceptor cells, bipolar cells, ganglion cells. Now, these different layers are the typical retina, is where you see. However, at a certain areas, especially a sensitive area, the macular region, the most sensitive area, the top two layers are pulled back. They're not missing. The cells are still there, but they're being pulled back, and so that the light can come down and interact with the rods and the cones. Pigmented layer, rods and cones, and you pull back the top two layers, the ganglion cells and the bipolar cells, so you have unimpeded view into the cells below. So here we can see the sclera, the cornea, conjunctiva, the iris or lens, anterior chambers, posterior chamber, aqueous humors in both of those, vitreous body, and then we have the retina, the choroid, the sclera, and right in through there where you have the top two layers pulled back, you have the macula. So you have the macula right there. But where the optic nerve comes in, you don't have photoreceptor cells, so as a consequence, you have a blind spot there. Here we can see blood vessels coming in through here. This is the optic nerve is coming in through there to the retina, and that really is the source of the retina. Here again, we can see how nicely the optic nerve is coming in. The blood vessels are coming in through there, making a plexus, and the optic head here is where you don't have photoreceptor cells, and indeed that would induce your blind spot. Choroid, very nice choroid here, blood vessels in the retina, which is very nice to see. In summary, we talked through about structures through which light passes, and we talk about structures that influence the iris, influence the image. The iris regulates light coming in and out, how much comes in. Ciliary bodies, ciliary processes secrete the aqueous humor, which maintains the pressure in the eye, and then also, which is the source of the pressure of the eye, and also the ciliary muscle contracts to regulate the focusing of the image on a photosensitive portion of the photosensitive cells. Now, the cornea itself is the one that focuses the image on the retina, but then it's the lens that does the fine focusing once it's there. So the fine focus is at the lens, but the coarse focus is the cornea. When you have reading glasses, you mimic your cornea. And in order to mimic the lens, you have to have bifocals for different distances. So we have the different structures of the eye. We have the embryologic changes that facilitate the eye that we have. The retina comes in through here all the way underneath the iris itself. The iris is part of the portion of the eye that came from the optic nerve. Optic nerve in through there, causing a blind spot right in through there, and we have the different layers of the retina. A couple questions. The integration of relationship among structures of the eye is produced and maintained by supporting layer of the eye holding pressure. Yes. So it's like pumping up a basketball. The structure is maintained by having a regulated pressure. A precise regulation pressure via aqueous humor production and outflow. So if the obstruction outflow, you may have glaucoma, but the trabecular meshwork prevents the outflow, or it regulates it. It doesn't prevent it, but it does reduce immediate outflow. Bony structure in the sclera of mammals, no. There are bony structures in some birds, but not in mammals, so the answer is A and B. Which structure is not correctly paired with its function? Iris regulates the amount of light that reaches the retina. True. The ciliary bodies alter the shape of the lens by the attached zonules. True. Trabecular meshwork enhances the outflow of the vitreous humor. No. The vitreous humor is in the vitreous body, and that does not flow. It's the aqueous humor that flows out. So that's wrong. The cornea focuses the image on the retina. Yes. The retina and choroid vessel provide nutrients to the retina. Yes. Which function least matches that of the retina, of the cornea? Protection? Yes. Is part of the structural layer. Support? Yes. Filter out undesirable light rays? It does. It focuses the image of photoreceptive portion on the photosensitive portion of the photosensitive cells? No. It focuses on the photosensitive cells, but not the photosensitive portion. That is a function of the lens. So the answer is D. We want to thank the various books that from which I was able to get images and drawings out to facilitate this lecture. Here is a picture of me in California, where I was playing at Stanford in the Senior Olympics. So that's the end of the medical school histology basic, the eye. Thank you.